Hello, everyone. My name is Tim Liu. I'm the CEO and co-founder at Senti Biosciences. Senti is a company that's focused on developing more intelligent medicines to overcome complex diseases. So at a glance, Senti was founded back a few years ago, raised a Series A in February of 2018, initially based out of technology from multiple different universities, including MIT and Boston University, focused on programming what we call gene circuits in living cells. And what these gene circuits allow us to do is create more intelligent medicines that can overcome difficult diseases with better drugs. So at Senti, what we're very much focused on is being the world leader in the development of these gene circuit therapies for smart cell and gene therapy products. We have developed a platform that allows us to create gene circuits that can be applied to a wide range of cell and gene therapy modalities. Internally, we're focused on allogeneic NK cells for oncology. That being said, there's many additional opportunities for collaborations to use these gene circuits in other modalities. We have a very experienced team that we've brought on board, including synthetic biology leaders, R&D team, CMC tech ops teams, and downstream development. We're fortunate to be supported by a great group of biotech investors, as well as strategic investors who have continued to demonstrate strong interest in what we're developing at Senti here. So just as a brief introduction, I started this company a few years ago together with Philip Lee, our chief operating officer and co-founder. His background comes from the manufacturing and technical operations side of the world. Kurt Herbert's our CFO and CBO, previously chief business officer at Sangamo Therapeutics, and he spearheads our internal finance and business development operations. Gary Lee brings 13 years of experience in the cell and gene therapy space, having taken 5 cell and gene therapy INDs into the clinic. So really key in terms of not only early stage research, but the translational biology that we need to do to get these products into the clinic. And finally, Jose Iglesias, our chief medical advisor, is really central in advising us on clinical strategy as well as our clinical development plans. So what do we do at Senti? We're very much focused on developing smarter drugs. And the idea is to be able to create dynamic drugs using our gene circuits that can be delivered into patients. And by doing so, interface with the complex disease biology that's there, rewire that disease biology, and treat those diseases in new and unprecedented ways. We are building a diverse pipeline of internal programs, all centered around using allogeneic NK cells as the modality for the delivery of these genetic circuits. Senti 202 is we call one of the first products that we're working on, which is adapting what we call OR gates and NOT gate logic to go after acute myeloid leukemia. The Senti 301 program is using what we call if-then logic to basically be able to deliver multiple therapeutic cytokines into the tumor microenvironment, but doing that under the control of a regulator gene circuit that we've designed. Senti 401 applies similar NOT gate logic to Senti 202, but is going after solid tumor indications. In addition to these wholly owned internal programs, we are very much interested and engaged with partnerships, working on using our gene circuits more broadly in other types of modalities, especially for non-oncology applications. So what are some of the key challenges that we seek to overcome with our gene circuits? We see that there are some central limitations with first generation cell and gene therapy products, even though these have been tremendously beneficial for patients and very exciting first movers onto the market. One of the key challenges we see is how to overcome the poor efficacy in some of these products due to the fact that they're typically point and shoot and only going after a single mechanism. And that can result in tumors that can escape due to antigen loss or due to the fact that we can't target the tumor microenvironment in a more comprehensive way. Number 2 is the challenge of trying to distinguish cancer cells from healthy tissues. Oftentimes with the current generation CAR T cells, for example, we're going after single target antigens, and we're relying on that single target antigen to uniquely differentiate tumor versus non-tumor cells. And so that becomes a key challenge if you can't find that single perfect antigen to go after. Number 3 is a challenge with toxicity. So if we want to arm cell therapies with particular therapeutic payloads, for example, that has been done before with IL-12, there is the chance that these payloads are expressed at too high of a level, and that results in systemic toxicity that you can't control very easily. And finally, there are challenges associated with toxicity, especially with systemic delivery of things like gene therapies that can result with very high level expression of transgenes in areas that you don't necessarily want them to be expressed. So we believe that all of these issues can be overcome with gene circuit technology and will allow us to take that next step in this field to really bring solutions to a broader swath of patients. So what are gene circuits? Gene circuits are essentially multi-gene elements that we can actually deliver into cells. And what these gene elements do is they basically introduce new programs into these cells. They allow us to design programs that sense different biological inputs, compute some sort of biological logic, and then translate that into a therapeutic output. So examples of some of the gene circuits that we've built in the company. So a NOT gate allows us to build cars that actually can avoid on-target off-tumor toxicity by essentially recognizing a don't eat me signal on healthy tissues. We can build what we call OR gates, which allow us to go after multiple tumor antigens simultaneously, or go after multiple tumor immunotherapies simultaneously to really maximize the efficacy of a product. There's the example of what we call if-then control circuit, where we can actually use small molecule regulators, FDA-approved drugs. If the drugs are present, then that triggers the cell to have a particular therapeutic activity. We also have what we call if-then endogenous control. So that's where we, for example, can design promoters that turn on only if a particular transcription factor is active in a cell type of interest, and only then express the therapeutic payload of interest. And finally, we have what we call the AND gate. This is where you can rely on two simultaneous disease signals to trigger activity of your therapeutic product, thus giving you greater selectivity for where that product is being active. So just to give you some examples of these types of logic circuits in action, this is an example of a NOT gate that we've built in NK cells. And so the idea here is if you have a particular target antigen that you want to go after, which is present on cancer cells on the left-hand side here, we want to be able to build an NK cell that can kill that. So the idea is we have a cell that actually is equipped with an activating CAR, can recognize this tumor antigen and trigger killing. However, if that antigen also happens to be present on some sort of healthy tissue, that becomes a big challenge because in those cases, we don't want to kill those healthy tissues. We want to spare those healthy cells and allow them to survive. So what we can do in those situations is identify what we call a safety antigen that is also expressed on those healthy tissues. And when that safety antigen essentially is engaged by this NOT gate CAR, that allows us to suppress the activity of the cell and preserve the survival of those healthy cells. So on the right-hand side, just showing you some data in demonstrating this, where we can trigger robust killing of a cancer cell that expresses a tumor antigen, antigen A1 in this case. But in the presence of the safety antigen, that engages the don't eat me mechanism, the NOT gate, and that minimizes the killing of these healthy tissues. And so this allows us to build therapies that are much more selective for the tissue type or the cancer type that you're going after. Number two is an example of what we call a OR gate. So in this particular case, we're actually building bicistronic CARs that are optimized for efficient dual antigen targeting so that they're able to activate in the presence of either antigen A1 or antigen A2, for example. And what we're showing you here in a particular model that's relevant to acute myeloid leukemia is when we have this A1 or A2 bicistronic CAR, we can enhance the killing of cells that express either antigen A1 or antigen A2 at high levels, or a 50-50 mixture of these cells compared to the individually targeting CARs, which are shown in these two particular graphs on the right-hand side here. Now, as an example of if-then exogenous control, that's again the idea that if in the presence of a small molecule drug, we then want to trigger the activity of a therapeutic product. I mentioned earlier, one of the areas that we're using this in is for treating solid tumors, where we want to use NK cells to try to drive the expression of therapeutic payloads in the tumor microenvironment. So what we've done is to design an artificial transcription factor, which we call ProDial. This binds to a specific promoter that can then activate expression of a downstream gene. In the absence of the small molecule drug, this transcription factor basically is inactive due to cleavage of the transcription factor by a protease, and that results in no expression of the transgene. However, in the presence of the drug, that inhibits that protease activity and allows the transgene to be properly expressed. What I'm showing you on the right-hand side here is an example of this working in vivo, where we basically implanted cells containing this ProDial circuit into mice expressing a therapeutic transgene, and then we dosed 3 different concentrations of this FDA-approved drugs, and we were able to show a dose-dependent increase in the amount of the therapeutic effector that you could detect in the plasma. So this essentially enables a physician-controlled dose-dependent protein expression in vivo and allows you to also control things like dynamics of gene expression. As another example, we've built that if-then endogenous control circuit. Again, the logic here is if a particular transcription factor or promoter is active in a cell type of interest, then you want the gene therapy to turn on and trigger downstream expression of a certain gene. So this allows us to create circuits that can actually respond to specific disease conditions. So as an example here, we designed a particular promoter that basically activated the expression of IL-10 in response to TNF-alpha in the context of a cell therapy. And again, we can show that with increasing concentrations of that TNF-alpha concentration, we can get a dose-dependent increase in secretion of that IL-10 molecule, allowing us to create a drug that can essentially sense and respond in vivo. Lastly, on the gene circuit side, we've built what we call that AND gate. The idea here for the gene therapy context is to try to link together 2 synthetic promoters, each one which is only active in a particular cell type of interest, and then only when both of them are active together do you output the gene of interest. So in this particular case, we are using this, for example, for cancer gene therapy applications, where you want to gate the expression of certain payloads to ensure that they're only expressed in cancer cells and not expressed in healthy tissues. So what we've designed here is an AND gate that requires both promoter 1 and promoter 2 to be active, and as a result, secretes a therapeutic protein, in this case, CCL21, which is a chemokine, a chemoattractant for immune cells. What we showed here is that we could actually transduce this gene circuit into 3 different cell types. 2 control cell types are dermal fibroblasts as well as healthy ovarian epithelial cells, in which basically the circuit is off. But in cancerous ovarian epithelial cells, we get about 10-fold or greater expression of the CCL21 payload in this particular context, again, giving us very selective expression of that therapeutic transgene. So all of these gene circuits ultimately come from the fact that we can design, build, and test, and learn from each experiment that we do. So at Senti, we have a core team of synthetic biologists that do all of the biological designs across our programs. We have individual project teams really dedicated to each therapeutic program that we're developing, complemented by a build team where we have an in-house viral core, an in-house cell core that can make viruses, make cells, transduce them, and then hand them over to our cancer teams to characterize in both in vivo and in vitro models. One important thing to note about this is the more times we go around this design cycle, the better we become at designing these gene circuits. And so there's a lot of synergy and efficiencies to be gained at Senti over time as we develop additional programs. So the remaining time, I just want to give you a highlight on how we're applying these gene circuit logic components to actual therapeutic programs. So I'll give you a highlight on Senti 202, how that's being developed, as well as for Senti 301, where we're using the if-then logic. In Senti 202, this particular program is really directly addressed against the need for better treatments in AML. As many of you well know, in acute myeloid leukemia, there's a lack of clean single targets that can create, as a result, creates toxicity risk when we use these products, especially when you get activity, off-target activity, on-target off-tumor activity against the bone marrow, for example. That limits the amount of drugs that you can actually dose into patients. In addition, treatment durability can be basically impacted by antigen escape in these systems, as well as the inability to target both blast and leukemic stem cells. So what we're building in this particular case is an engineered cell therapy containing our logic gates that target multiple tumor antigens in AML and have the potential to cure patients in the absence of that bone marrow transplant. So the way this particular system works is we actually have built a logic-gated CAR-NK cell that contains 3 different CAR receptors. 2 of them are activating receptors that target either antigen A1 or antigen A2, and thereby broaden the spectrum of killing of the AML cells, both on leukemic blasts as well as on leukemic stem cells. In addition, we've identified a safety antigen, which we call SA1, which is highly expressed on healthy hematopoietic stem cells, but basically undetectable at very low levels on any AML cells of interest. So this particular safety antigen serves as a NOT gate, a don't eat me signal to the cells, and basically prevents the activity of our CAR-NK cells against those healthy tissues. This allows us to spare the healthy bone marrow and to dose these patients aggressively with this logic gated circuit so we can hit the on target, on tumor antigens in a very strong fashion to maximize efficacious use cases here, as well as to allow us to develop this program for patients that don't qualify or otherwise can't find a bone marrow transplant. Senti 301 is a program where we're applying the small molecule regulator dial, the if-then logic, to the treatment of solid tumors, including liver cancer, where we have an immunosuppressive solid tumor microenvironment that limits the efficacy of existing immunotherapy approaches. So in this particular cases, we're engineering, again, our CAR NK cells to go after specific tumor antigens, but we're also engineering those cells to contain therapeutic payloads that can be expressed in that tumor microenvironment to overcome immunosuppression. So what we've done here is identified specific combinations of therapeutic payloads that can be highly expressed in the tumor microenvironment to trigger an anti-tumor response, for example, by recruiting both innate and adaptive arms of the immune system. And all of this is being done under the control of that ProDial regulator, where we can use small molecule drugs to either control the timing, the dosing, of the expression of these therapeutic payloads in vivo, thus enabling enhanced efficacy and enhanced safety. All these programs, as I showed in this particular graph, are just the tip of the iceberg. We're going after initial targets that we believe are well clinically validated and represent low-hanging fruit for the gene circuit technologies. That being said, we can readily expand both the OR gates and the NOT gates, as well as the regulator armed cells, the if-then armed cells, to a variety of additional targets and additional cancer applications. So we're very excited about the opportunity here to build a multi-product portfolio downstream. At Senti, we have been establishing a manufacturing expertise internally, as well as facilities to support early stage process development and CMC, along with processes that we believe will be scalable and translatable to future GMP manufacturing, which we intend to internalize and bring in-house. So finally, I just want to briefly mention, how we view the gene circuit technology more holistically. Gene circuits are essentially the software that go into a variety of cell and gene therapy modalities, which we term the hardware. So these include different cell types, including NK cells, T cells, iPSCs, and other stem cells like mesenchymal stem cells. And we've worked with many of these different cell types and have shown that these gene circuits can be functional in that context. Similarly, we can view gene therapies as a modality. So whether we're talking about AAV, lentivirus, or non-viral vectors, these can all be enhanced in terms of their selectivity as well as efficacy with our gene circuits. And so although Senti is internally focused on oncology for our initial therapeutic use cases, we are quite interested in using these gene circuits in additional modalities and outside of oncology, and are open to partnering discussions in those particular contexts. So just to end here, Senti strives to be the world leader in the development of gene circuits for these next generation smart cell and gene therapy products. We've built a robust pipeline based off of our gene circuit platform that includes both wholly owned internal programs focused on NK cells for oncology, as well as potential collaborations in additional modalities. We have a team that can execute along the entire spectrum of the development activities, ranging from early stage R&D to CMC tech ops to downstream clinical development. And so with that, happy to end there and take any questions. Thank you.